Hello everyone, my name is Vidal Yahya and today we're gonna explore together bacteria. We're gonna talk about them in a particular way. So, for sure you must have heard about some really dangerous bacteria, responsible of terrible disease and even death. We must know that bacteria are like knives. They are really dangerous, but they're as well extremely useful for our body's daily activities. So, let's go together to explore the relationship between our bodies and bacteria. By the way, do you know this guy? His name is Richard Jefferson. He is a famous American molecular biologist. Richard Jefferson in 1994 first articulated the Hologenome Theory of Evolution.
The hologenome theory is a postulate put forth in 2007 by Eugen Rosenberg and Dilana Zilber-Rosenberg, stating that the object of genomic natural selection is not a single organism, but the organism and its microbial communities.
Human beings are ecosystems on two legs, each of us carrying enough microbes to outnumber our human cells by 10 times, and our genes even more. Every living species is the result of a complex evolutionary history that depends on many factors, not least the influence of microbial forms of life that live in close contact with it. Darwin's selection does not merely act on the host, but on a complex biological system made by the host and its microbial communities, the hologenome. Bacteria can deeply influence many aspects of our lives, from cerebral development to digestion, from defense against infections to our well disorders. Well, the research on the human body is evolving really fast. Perhaps, we don't know. The role of the microbiota, the commonly known gut flora, is not their direct contribution on the human body, but rather the protections that they offer against the growth of other pathogens. Pathogens such as the Clostridium difficile, an opportunistic pathogen of the intestinal tract causing chronic and debilitating diarrhea in patients whose gastrointestinal flora have been altered by the administration of certain antibiotics. So, the genomes of host-dwelling microbes must be taken into account along with that of the host when evaluating environmental selection pressures. In addition, recent research by the Rosenberg lab suggests that the commensal bacteria play a role in mate choice by the fruit fly, the Drosophila melanogaster, further supporting the hypothesis that greater genetic trends are determined by symbiotic microbiota. Last summer, I had the great opportunity to lead 3 weeks of scientific research at the prestigious Weizmann Institute in Israel. There, I worked on the importance of flies' gut microbiome in coping with stress. The Drosophila is a really spread animal in many labs. Why? Because it is perfect for many researchers due to its short life cycle, its great fertility, its defined microbiome, and its simple genome compared to the human one. So, we were talking about the importance of the fly's gut microbiome in coping with stress. But what is a stress? Let's see it. A stress can be a change in the ecosystem, in temperature, a new predator. It might be the lack of food or this administration of hardly digestible food. The stress can be faced in 2 ways. Firstly, the genetic mutation. But it's really hard. Most commonly, a stress can be faced with the microbiome adaptation, isn't it? I think it is. How could we then draw out some conclusions about the importance of microbiome? Simple. We just compared how the flies reacted with and without bacteria to a stress that we have chosen. So, in order to compare a reaction to a stress and to a normal situation, we need both stressful situations and a normal situation. So what I have done was given the flies 5 different kinds of food. One was the normal food, winning the control experiment. This normal food was made of Polenta valsugana and yeast. Then, the other 4 were unusual foods, which were hard to digest because these flies don't have the right enzymes. So, 2 were alcohols, the xylitol and the sorbitol. One was the cellulose, and the last food was the fruit-based food. So, the flies were divided into 3 groups. One was with their original bacteria, we didn't touch them. The second group was germ-free, it didn't have any bacteria. And the third group was with bacteria that did not originally belong to that group, but to other different animals. So, this way we will be able to understand what is gonna be the importance of the microbiome on the fly's reaction. I have measured the influence of the bacteria on my flies according to 2 criteria. Their health, measured with the time that it takes them to develop, and their survivability rate. So, first of all, I have measured their growth speed. How fast did the eggs stretch and become pupas? The data I collected clearly shows that the flies that had bacteria became adult in 7 days, while it took around 3 days longer to become adult to the flies without bacteria. So, the flies without bacteria, the germ-free flies, were much slower than the normal flies. Then, I compared the number of flies that survived to the 5 different foods with and without bacteria. So, what can we see is that the germ-free flies survived only with normal food, while the flies with bacteria managed to survive the stressful food and became pupas. But finally, the last experiment gave a sensational result. While the flies with their own bacteria did not survive to xylitol and just few of the Drosophila managed to live in the fruit-based food, the flies that borrowed other bacteria, which did not belong to their original microbiome, did survive with a great percentage, much better than the normal flies. So, this last experiment showed us a sensational fact. Even other bacteria can help us to adapt to stressful situations. Our thoughts on the relationship between us and the bacteria are changing quickly. The 2007 launch of the Human Microbiome Project by the National Institute of Health has reinforced the idea that microbes are an indispensable part of human health. But whether this principle will be extended to encompass all of evolution remains to be seen. Certainly, recent research on the effects of microbial population on the fitness and mating preferences of other hosts must be taken into consideration when evaluating the legitimacy of the assertion that it is the holobiome and not the individual organism that is the object of natural selection. Did you understand now? For example, without bacteria, even eating this tiny banana would become almost impossible for us, human beings. So guys, I hope you have appreciated my short lesson on the Hologenome Theory of Evolution, and I wish to see you really soon. Bye bye!